So now we're going to discuss about all the important images that you have to remember before you appear for the exam. Now, what are the important images which are very relevant to the clinical history that will also be presented? Those are the important ones that I'm going to discuss today. So this is about 25 images that we will discuss today, and this will all be in less than 30 minutes. So this is a particular image that you have to always remember, because this is something which is considered to be one of the important images for the exam, and this is known as your Koplik spots. So Koplik spots is something that you have to remember when we are talking about measles or rubeola. Now, what is important for you to know about this particular finding is this is seen in the buccal mucosa. It is bluish spots which are seen, and this is considered to be something which is always associated with measles. Now, there's another important spots that you have to remember, which is also very close to Koplik spots, which is called as Forchheimer spots, and that you see in German measles. So please do not confuse both. Koplik spots are seen in rubeola or measles, whereas Forchheimer spots are seen in German measles. To the next important image, can you identify the type of staining that you see here? So what is this particular mount called? It is called as LPCB mount, where L stands for lactic acid, P stands for phenol, and the dye is cotton blue. The role of lactic acid is it maintains the morphology. Phenol acts as a disinfectant. And you will see that cotton blue is considered to be the dye. So this is lactophenol cotton blue, and you see a lactophenol cotton blue mount from a culture. So after you do a culture, you will be able to appreciate this, and LPCB is mainly done to identify the fruiting bodies. Now, can you identify this particular image that you have, where you have multiple ones which you see? What are these images of? Can anybody tell me what are these images of? Can you identify the image? Can you tell what exactly are we trying to show you in this image? Now, this is in fact LPCB mount of your dermatophytes. And how do you remember, or how do you identify your dermatophytes? It's based on your macroconidia and microconidia. So how do you remember this? Remember this as TEM, T-E-M, where trichophyton, epidermophyton, and microsporum. And macroconidia, how do you remember this? Please write R here and R here. This is followed by N here and N here. So this is how you will write, and there will be A here. So how do you remember this? Trichophyton, you will see that your macroconidia are rare, and if they are present, what is the shape of the macroconidia? It is pencil shaped. In epidermophyton, N stands for numerous. And what is the shape of this? It is club shaped. And when it comes to microsporon, here again N stands for numerous. And what is the shape? it is spindle shaped. A stands for abundant. So you will see abundant microconidia when it comes to Trichophyton species. In Epidermophyton, A stands for absent, whereas R stands for rare. So please remember, this is how you should remember, and if you can see here, this is a species of Trichophyton. Now it is Trichophyton because you can see abundant of your microconidia, and you can see that there is pencil shaped macroconidia. Now coming to your second one, here can you see microconidia? You cannot see microconidia, whereas you can see numerous club shaped macroconidia, so this belongs to Epidermophyton. And the last one to remember is microsporum, where you will see that the macroconidia are spindle shaped and you can see rare microconidia. So this is how you would be able to remember all of them. So always remember, this is the order to remember for the exam. Please write it down as STEM with macroconidia and microconidia. Start off with R on the beginning, R here in the end, N and N here, A and A above. So that's how you will be able to remember the shapes of it. So this is important for you for the exam. So you should always remember that this is the appearance that you will be able to identify based on your LPCB mount. To the next image, can you identify this particular image here? This image of what is known to be as vesicular lesions. Vesicular lesions involving your dermatome. And this is nothing but your herpes zoster, or this is also known as your shingles. So herpes zoster or shingles, so this is what you have to remember. For this particular image where you can see that there is an involvement of dermatome. So vesicular lesions involving dermatome is something which is very consistent with herpes zoster or shingles. This is because of latent infection of varicella and reactivation which can occur because of immunocompromised state or because it is something which is commonly seen in old age. So please remember this particular image for shingles. Can somebody identify this particular image? So if you can see, this is Enterobius vermicularis. This is the egg of Enterobius vermicularis. And what is it referred to? What are the names of Enterobius vermicularis? What are the various names that you have of Enterobius vermicularis? So Enterobius vermicularis, can somebody tell me what are the various names that you have? What is Enterobius vermicularis also called as? Is there anything that you have to remember about Enterobius vermicularis? What is it also called as? It's called as pinworm or seatworm. And what is the main important symptom? It presents with perianal itch. And when it comes to the egg, you can see that it is plano-convex. Plano-convex egg which you can see for Enterobius vermicularis. Can you identify this particular image? Can you identify this particular image that you see in this? Now, this is an image of what is known as Anaerobic magnifi. This is an image of Anaerobic magnifi, and what is important for you to know about Anaerobic magnifi. Now, Anaerobic magnifi is the only Penicillium species which is dimorphic. So, Penicillium as such is not dimorphic, it's a common lab contaminant that you see, but Anaerobic magnifi is a dimorphic fungi. And how does it exist? So, Anaerobic magnifi exists in the form of yeast and it exists in the form of mycelium. And what do you see here? It is yeast with central septa. And when it comes to mycelium, it produces diffusible red pigment. So, diffusible red pigment is something that is associated with Anaerobic magnifi, and please remember this is one of the important image based questions that are usually asked when it comes to pigment producing fungi. So, Anaerobic magnifi also is nothing but the new terminology for Penicillium magnifi, and always remember the reservoir is important. It is bamboo rats, and in India, epidemiology wise, it is common in Manipur. Around the world, it is common in Southeast Asian countries. So, this is in fact what you have to remember when it comes to Anaerobic magnifi. Now, can you identify this particular image? What is this particular image trying to show you? What is this particular image trying to show you? So, this particular image is that of what is known as, exactly, very good, it is chrome agar. Now, chrome agar is used for identifying species of Candida. So, species of Candida are identified by chrome agar. The different species of Candida gives different colors. So, you will be able to know if it is Candida albicans, Candida tropicalis, Kiefer or Vishwanadi based on the different colors which it produces. And this is basically because of the different metabolites which are involved and the different kind of colors that it produces, which helps in identification. So, this is a very important image that you have to remember. Now, can you identify this particular media where you are seeing jet black colonies? What is this particular media that we are showing here which is having jet black colonies? Somebody tell me what gives jet black colonies. Very good. And this particular media is Wilson and Blair media. So, Wilson and Blair medium, which is giving you jet black colonies. Very good. Can you identify this particular medium which is used for certain anaerobic organisms. Can you identify the medium which is shown? What is this particular media which is shown here, which is used for certain anaerobic organisms? There are a few questions that are also asked. I will try to answer a few questions which you are asking me, how to avoid overthinking while studying. So we'll probably have the last 2 minutes just to discuss about all of that. I'll tell you how to not overthink. Now coming to this particular medium, it is called as Robertson cooked meat broth medium. It's an RCM medium which is used, and if you can see here, it is the particles that they ask you, it is beef heart. So these are used as the particles which are present in the meat particles. And what do you study in RCM? You study for Clostridium, where Clostridium tetani produces proteolytic change. So you can see that the meat particles turn black in color. So that is the importance of Robertson cooked meat broth medium. Very good. So now they've given you an image where they have shown you what it looks like a KOH mount. So this is a KOH mount, and if you can notice here, they are trying to show a particular type of finding here, which is nothing but aseptate hyphae. If you can see here, it is aseptate hyphae. And if it is aseptate hyphae, what are we thinking about? So this could be a case of Zygomycosis. This could be a case of Zygomycosis where a person presents with aseptate hyphae. And what are the typical features of your Zygomycota group? So Zygomycota, the important organisms to remember can be RAM, where R stands for Rhizopus, A stands Rhizomucor, A stands for Absidia, and M stands for mucor. So KOH mount with aseptate hyphae could be a case of Zygomycosis. Whenever we are talking about mucormycosis, this is what is this typical presentation. Now aseptate hyphae, angio-invasive in nature, something that is very, very consistent with mucor and Rhizopus. And what is the typical culture finding that you see in Rhizopus and mucor? It is called as salt and pepper appearance. And these are also known as lid lifters. So it looks like the growth is such that it can lift the lid of the petri dish. So this is called as salt and pepper appearance, or these are known as lid lifters. Now what is this particular image that you have to remember? Now this is an image of what is known as Giardia lamblia. So Giardia lamblia. And it is the motility that is very important for you to know. What is the motility of Giardia lamblia? Giardia lamblia, the motility is considered to be as falling leaf motility. Falling leaf motility is very important for you to remember for Giardia lamblia. And when it comes to the clinical history, please remember this is responsible for fat malabsorption. Giardia is responsible for malabsorption. Very typical finding that you see, very good. It is falling leaf motility. And clinical history, the most significant one that you have to remember, especially in pediatric age group, there is fat malabsorption that you see. Can you identify what is this appearance? What is this appearance known as? Can you tell me what the appearance is? So this appearance is known as slapped cheek appearance. An appearance which is very significantly seen. And where do you see slapped cheek appearance? Very good. Slapped cheek appearance. Where do you see it? What causes slapped cheek appearance? Which virus is responsible for slapped cheek appearance? Very good. The slapped cheek appearance is caused because of parvovirus. Can you identify the type of staining that you see here? What is the type of staining that you see? What is the type of staining that you see here? So this is the appearance that you have to remember for modified acid-fast staining. It is modified acid-fast staining which is done, and this is the organism that you can see here is Cryptosporidium parvum. So to isolate or to identify the oocysts of Cryptosporidium parvum, you do modified acid-fast staining. It's also called as Quignon's method, where it's known as cold staining. Now in modified acid-fast staining, the change that you do here is your percentage of H2SO4, where you use 1% of H2SO4 for staining. So this is to demonstrate the oocysts of Cryptosporidium parvum. So oocysts of Cryptosporidium parvum are demonstrated by modified acid-fast staining. Can somebody identify what this appearance is? So you have to tell me what the appearance is. So this is called as, so there are 3 C's that you have to remember for this condition, which they ask even in the exam. One, it is the condition chromoblastomycosis. So chromoblastomycosis. The other C to remember is cauliflower-like growth. So this is usually described as cauliflower-like growth. And the third important one to remember is this typical appearance that you see, which is called as copper penny appearance. Or it's also called as sclerotic bodies, medullary bodies, muriform bodies. So these are all the various names that are used to describe this typical appearance, which is called as copper penny appearance seen in chromoblastomycosis. Chromoblastomycosis and phaeohyphomycosis are mainly differentiated by the absence of your copper penny or your sclerotic bodies in phaeohyphomycosis. Now this particular test, so it's very important for you to identify a few preliminary tests which are done for identification of the organism. This particular test is known as your oxidase test. So oxidase test, this is an oxidase positive. The most important organism that you have to remember for oxidase test is Pseudomonas. So Pseudomonas aeruginosa is an oxidase positive organism. Very important for you to remember for the exam. Now this is the image that you have to remember, where you can see a terminal spine. What exactly do you have to know in your terminal spine? This is in fact the image of egg of which particular organism? Very good. Egg of Schistosoma hematobium. Can you identify this? What is this? Is it a tick? Is it a mite? Identify this particular one. So this is nothing but a louse. So this is an image of louse, and what is important for you to know, this is responsible for relapsing fever, epidemic relapsing fever, and it's also responsible for pediculosis. So this is basically a louse. The louse causes your relapsing fever. Can you identify this particular appearance? So this appearance is spaghetti and meatball appearance. Where you see broad hyphae with yeast, broad and short hyphae with yeast, which gives a typical appearance which is called a spaghetti meatball appearance. And where do you see this? This is seen in Tinea versicolor. And this is the typical appearance that you see, which is called as fried egg colonies. Fried egg colonies in SDA with overlay of olive oil. And this is caused by Malassezia furfur and Malassezia globosa. Can you identify the type of microscopy that you see? What is this type of microscopy that you see here? So what are we trying to demonstrate in this image? It is what is known as your dark field microscopy. So this is dark field microscopy. where you are actually demonstrating your spirochetes. So this could be Leptospira, which is in fact what you can identify by dark field microscopy. So these are very thin organisms, many a times not something that can be visualized unless you do a very good. It's a dark field microscopy unless you do a silver impregnation. Many a times these are so thin that it cannot be visualized. So dark field microscopy, which is done for identifying spirochetes. Can you identify this particular antibody that you should know, which is pentameric? So this is a pentameric structure. So which is this particular antibody that you have to remember? Very good. It is pentameric, it is IgM antibody. And most common antibody be elevated in acute infections. It's IgM. Coming to the next image that you can see. So here you can see that there is infection of the, so this is something which is commonly seen. It's a ear infection that you can identify. So here this is an image of what is known as malignant otitis externa. Malignant otitis externa. The most important organism that you have to remember is Pseudomonas. So Pseudomonas aeruginosa is responsible for malignant otitis media. What are the other conditions that you have to remember for Pseudomonas? Other conditions which is responsible, which it causes are, it causes what is known as green nail syndrome. It can cause Shanghai fever. Ecthyma gangreosum. Most common organism to cause infections in burns. Green nail syndrome, Shanghai fever, ecthyma gangreosum, and most common organism to cause infections in burns. So this is Pseudomonas. So it is malignant otitis externa caused by Pseudomonas. So what is this particular image? So this particular image that we are trying to show you is also modified acid fast staining. And the organism here, what is the organism here that you have to remember? Here, if you can see, it is branching, gram-positive branching filamentous bacilli. So this could actually be nocardia. So nocardia is gram-positive branching filamentous bacilli. And what is the important test that you have to remember for nocardia? It is called as paraffin bait technique. So paraffin bait technique is something which is done for nocardia, and please remember it is a test where you can identify if it is nocardia or not based on the ability of nocardia to use paraffin as a sole source of carbon. Now this is an image of what is known as Castaneda's biphasic medium. So Castaneda's biphasic medium, where you have a solid and a liquid medium. And what is the advantage? So the advantage of biphasic medium is reduces contamination during subculture. And lastly, it is used in which organisms? Salmonella and most importantly, Brucella. So you use it for Salmonella as well as Brucella. So this is an image of LJ medium, which is used for tuberculosis. And what is the important finding that you have to remember for LJ medium? It is rough, tough and buff colonies are seen on LJ medium. Rough, tough and buff colonies on LJ medium. And how do you sterilize your LJ medium? It is by inspisation. So you use LJ medium is sterilized by a method which is called as inspisation, and this is the typical colony finding that you see. Now this is an image where you are trying to show what is known as your Donovan bodies. So this is an image of Donovan bodies, and what is the organism that you have to remember for Donovan bodies? Anybody can tell me what is the organism? Organism that you have to remember for Donovan bodies. This is a case of Donovanosis. Can you tell me which is the causative organism for it? It's not chlamydia. What is the causative organism for it? Donovanosis is caused by Klebsiella granulomatis. Very good. Klebsiella granulomatis. Causes Donovan bodies, and a typical finding could be beefy red ulcer. And on demonstration of Donovan bodies. And the last image to remember, now what are we trying to demonstrate in this particular image? So what we are trying to demonstrate in this particular image is whenever you have an antigen, you will see that the antigen presenting cells along with MHC class 2 presents the peptide antigen to your T cell receptor, or which is present on your T cells. Now here what are you trying to demonstrate? Here you are trying to show that this is nothing but a super antigen. Why is it a super antigen? Here it is a super antigen because you can see here that it's directly going and binding to the variable beta region of your T cell receptor. So variable beta region of T cell receptor. Because it binds to the variable region of T cell receptor, it actually causes massive release of your cytokines, and because of the release of cytokines, it can also cause a massive release of your antibodies, causing hypergamma globulinemia. So super antigen is something that you always have to remember as something which bypasses your antigen presenting cells, your MHC2, and directly goes and binds to the variable beta region of your T cell receptor, causing massive release of your cytokines. So these are all the important images that you have to remember before you go for the exam, because many of these images have been previously asked in the exam, and these are of great importance. So any questions? Very good. Yeah, super antigen is by staph and streptococci. You do see it with staph and streptococci, along with Yersinia. Do you have any questions that you would want to ask? One of the questions that was asked is, how do I prevent overthinking in the exam? Guys, it's very, very simple. You will overthink whenever you have too much of a doubt about one or two options. Now, the only way to not overthink is to be sure of the answer. The only way to do that is revisions. The more you revise, the more points you will cover, the more easier it becomes in the exam. So overthinking can only be overcome by, repeated rapid revisions and constantly discussing it. I always suggest that, when you're reading with others, it becomes, something that you are also telling yourself. Somebody is also helped by it. Most importantly, it also gives you an idea, if you remember it or not. So always revisions are very, very important before the exam. So these are all the important images. Any questions for the last 1 minute? So how many questions, how many MCQs should I solve per day? See, there's one approach, guys. The number of MCQs that you have to do in a day should not be less than, maybe 50 or 60 in a day. But having said that, do not, the only main idea about solving these questions is the more you solve, the more it widens your horizon, more chances of the questions coming from it. So there is the minimum that you get, the minimum that you have to do is not less than 50 or 60. Thank you so much. So must do 10 topics before the exam date. Maybe I should make a video of all the must do 10 topics before the exam. We will do that as well. How to revise micro in the last final month? So there are, the best way to do it is revise all the important points, probably many of which you have noted during your your study period. So it's the important points that you have to revise in the last final month. Thank you so much. Thank you. Do that. We'll do the last 10 topics that you should know before you go to the exam. We'll do that edition for DocTutorials. Thanks a lot, Narsimha. Thanks a lot. Thank you, guys.